Good morning, all of you. Once again, I welcome all of you to this botany webinar session. Earlier, we were discussing the Unit 1, Chapter 4, Plant Kingdom. As part of the plant kingdom, in the previous class, we discussed about the algae. In algae, we discussed about the three classes of algae: Chlorophyceae, Phaeophyceae, and Rhodophyceae. We discussed about their important characters, their features, their role in our life, etc. In today's class, we will be discussing about the bryophytes and pteridophytes. Bryophytes are the first amphibians of the plant kingdom, and pteridophytes are the first terrestrial plants with vascular tissues. Today, Mr. G.SATHYANARAYANA garu, PGT Botany from TSMS Bandariguda, sir will be discussing about these topics. In the next session, he will conclude. So I request all the students to cooperate and coordinate and have a good command on the subject by listening to the sir classes. I thank Manjari madam and all the officials who are helping us in conducting these webinar sessions. Thank you. Satyanarayana garu.
Yeah, okay. Thank you so much, sir.
Okay, screen share cheyandi.
So, respected principals and coordinators, dear children, very good morning. In this session, last session, we learned the Unit 1, Chapter 4, session number 1, that is the classification of plant kingdom. In plant kingdom, we learned about the alga. So in alga, we completed the three classes: Chlorophyceae, Phaeophyceae, and Rhodophyceae, and we also learned their economic importance also. In this session, we are going to see the another important concept of plant kingdom, that is bryophyta. So children, once again, if you recapitulate our plant classification, the plantae have classified into two subkingdoms, cryptogams and phanerogams. We already know what is cryptogam and what is phanerogam. In cryptogams, again, divisions are divided. So thallophyta, bryophyta, and pteridophyta are divided, and in phanerogams, spermatophyta is there. So in last session, we completed the thallophyta. So in thallophyta, we learned about the alga. Now in this session, we'll see the another cryptogam, that is bryophyta. So the bryophyta, as Sar said, bryophyta are the first amphibians of the plant, and the embryo, the development of embryo is also starting from the bryophytes only. In alga, we could not find the word embryo, but in bryophyta, when it comes to it, the in bryophyta, the zygote will divide it. In pteridophyta, the embryo become formed. In bryophyta, the zygote will divide it and it produces the sporophyte only. So embryo, the first embryo is formed in pteridophyta, not in bryophyta. So in pteridophyta also, in today's session, we complete pteridophyta also. So bryophyta are the first amphibian, that means first land living plants, or we can call amphibians of the plant also. For their reproduction, they require water only. When it comes to pteridophyta, the pteridophyta are the first vascular cryptogam plants. So the vascular system, that means the xylem, phloem, are developed in pteridophytes. And the embryophytes also, that means the embryo, the seed habitation is started from pteridophytes. And the embryo development is also taken from, starts from the pteridophytes only. So in today's session, we'll complete these two divisions, bryophyta and pteridophyta. So let us see the bryophytes. Now, if we see their general characters, the bryophytes, the plant body is called thallus. They are the archegoniates. That means archegoniates means the sexual reproductions have archegonium and antheridium. And embryophytic. So embryophytic, these are the first embryophytic plants. So from here, pteridophyta and gymnosperms, angiosperms, they also having the embryo. So they are the first embryophytic and atracheophytic cryptogams. So atracheophytic means here the vascular system is not developed. Xylem, phloem, the conductive tissue is not developed. So they are the first embryophytic, atracheophytic, and cryptogamic plants. We see that generally they are called as amphibians of the plant kingdom. Generally, they lives on the soil. but the habitation must be contain the water. That means the habitation must be moist or wet conditions. So for their reproduction, they require the water. So they survive in with the help of the water. They survive. They are living on the soil. So that we can call them as amphibians of the plant kingdom, and they are the primitive land plant. So in when it comes to the alga are mostly aquatic habitat. But in case of the bryophytes, they are the first land plant. So first are primitive land plant, and they play very important role in succession of the soil on rocks, soil substitution. That means the formation of the soil. If you take the example of sphagnum peat is one of the important bryophyte which is useful for the succession of the soil. To form the soil, they are required. So economic importance also there. First, primitive land plants, atracheophytic and embryophytic plants have placed in bryophyta. Now, if we see their structure, the plant body, it is called thallus. We already learned what is thallus. The thallus is undifferentiated plant body. That means we cannot differentiate what are the root, stem, and leaf. So the plant body is thallus, and it has the unicellular or multicellular rhizoids. Now, if we see rhizoids, generally the thallus is also having the dorsiventral surface also. When it comes to the the Markensia body having the dorsiventral part. So at the dorsal part, the rhizoids are present. Now, how the rhizoids are present? The rhizoids are made with the unicellular or multicellular. In Markensia, the rhizoids are made with the unicellular. But when it comes to the Fineria and Polytrichum, the rhizoids are multicellular. So unicellular to multicellular rhizoids are present. Now we'll see what is their function also. What is their function? They are useful to attach to the substratum. Any base or any rocks or any other substratum, if it is there, to attach with the substratum, the rhizoids are present. Apart from the rhizoids, some bryophytes have also having scaly leaves or scale leaves like structures also present. They are also helpful for the attachment of the substratum, attachment with the substratum, and to observe the water from the soil also. So the plant body is thallus. On the thallus at the dorsal side, the rhizoids are present. Now the other part are present at the ventral side. Now what are their other parts? See here, we already learned the mosses, or we can commonly call the bryophyte. Bryophytes process root-like structures called rhizoids. Now these are the rhizoids. They are the root-like structures, but they are not exactly root. They do the function of attach with the substratum and to take the water and mineral from the soil only. And leaf-like structures called phylloids. So these are the leaf-like structures. They are called phylloids. So in the phylloids, if we take the TS of the phylloids, in there we can find the chlorenchyma tissue. Mostly they have the parenchymatous tissue. In parenchymatous tissue, one of the type is chlorenchyma. So chlorenchyma and arenchyma tissues are present in the phylloids, so that it shows the photosynthetic process. So they are the autotrophic plants, and they have the phylloids. Within the phylloid, the chlorophyll tissue is present, and it exhibits the photosynthesis process. And stem-like structure called colloid. So a stem-like structure is present, it is called colloid. So in plant body, the rhizoids are present, the leaf-like structures, phylloids are present, and stem-like structure, colloid is present. And if you take the TS of, and if you observe the plant body or the TS of the plant body, generally the plant body is mostly made up of the parenchymatous tissue. Parenchyma is a soft tissue. So in parenchymatous tissue, the chlorenchyma, arenchyma is present. In bryophyte, the chlorenchyma and arenchyma mostly present in phylloids, and some of the bryophytes in their rhizoids also the chlorenchyma tissue is present. Now the main plant body of bryophyte is haploidic in condition. Haploid means only a set of chromosomes are present. So the dominant phase of the bryophyte is gametophyte. Children, you can see here, this is the gametophyte or main plant body, which having the rhizoids, colloid and phyllite. This is the main plant body. On the main plant body, the partially dependent or totally dependent one more stage is present. This stage is called sporophytic stage or sporophyte. This is a sporophyte. So the sporophyte is partially or completely depend on the gametophyte. This is the main plant body which is called gametophyte. It is haploidic in condition and the sporophyte is a diploidic in condition. So in this image, it's a funereal image. So here you can see this is the sporophyte and this body is the gametophyte. So the main plant body is what? Gametophyte. And what is the condition? It is haploidic in condition. That means only single set of chromosomes are present. So two types of plant bodies are present, gametophyte and sporophyte. So morphologically they are different. See, if you see morphology of the gametophyte, it has a root-like structure, leaf-like structure and stem-like structure. But when it comes to the sporophyte, it do not have any structures. Only capsule is present. Within the capsule, the spore mother cells are present. That means morphologically they are different. So this kind of condition is called heteromorphological condition. Morphologically they are different. So heteromorphological condition. So two types of plant bodies are present and the main plant body is called gametophyte. The sporophyte is partially or completely depend on the gametophyte. Now the sex organs, when it comes to the sex organs, generally in bryophyte, the sex organs are multicellular, jacketed and stalk-like structures or stalker. In alga, the sex organs are unicellular. But in case of bryophyta, the sex organs are multicellular and they are jacketed like structures. See here, within the multicellular structure, we can find neck cells, egg cells, and the cap cells also. So 8 to 16 cells are formed in to form the sex organ. And what are the sex organs? The male sex organ is called antheridium. So this is the antheridium, the Markensian antheridium. The antheridium or archegonium or the sex organs produced at the tip of the thallus. Okay, so here it is called antheridiophore. It is a stalk-like structure and it bears the antheridium. Here you can see the antheridium. So antheridium is a male sex organ and it produces biflagellated antherozoids. Within the antheridium, the spores are produced. These spores are called antherozoids. So if you see the structure of the antheridium, now within the antheridium, see it is the antheridium. So a multicellular stalk is present. This is a multicellular stalk, and within the on the stalk we can find the antheridium. Within the antheridium, the antherozoids are produced. So the antherozoids are the male sex organs. Now when it comes to the female sex organ, the female sex organ is called archegonium. This is the archegonium and this is called archegoniophore, which bears the archegonium. Generally the archegonium is flask-shaped condition. See, if you see this one, it is a flask-shaped condition, flask-like structure. So within the archegonium, we already learned they are the multicellular organs. So in archegonium, we can find one egg cell and 8 to 10 neck canal cells. So these are the neck canal cells. One egg cell is present and 8 to 10 neck canal cells are present. So the condition is multicellular. Overall, 18 to 16 cells are present in archegonium. And what is the shape of archegonium? It is flask-shaped condition and it produces a single egg and other neck cells also present. So the female reproductive organ is called archegonium. Within the archegonium, one egg cell only present. The male sex organs are called antheridium. Within the antheridium, many antherozoids are produced and the antherozoids have the flagellates. Two flagellas are present. So they are biflagellated antherozoids are produced. The antherozoids released into the water. So the antherozoids released into the water. With the help of the water, the antherozoids reaches to the egg. So that for their reproduction, they compulsory require the water. Okay. So antherozoids, when fuses with the egg cell, they forms the zygote. So this kind of sexual reproduction is called zooidiogamous oogamy. So it is a zooidiogamous oogam. Zooidiogamous means the motile spores, the movable spores are present, because the antherozoids have two flagellas. No, with the help of the flagellas, they are moving. So if they move, they call zooidiogamous. Okay, so zooidiogamous oogamy is observed in bryophytes, and here they do not produces any tube-like structures. In zooidiogamous oogamy, they do not produces any tube-like structures. When it comes to the higher plants, the pollen grain produces the pollen tube, but here any tube-like structures are absent. So that this type of condition is called zooidiogamous oogamy, and it's a life cycle. So this is a Markensia thallus. It produces the female gametophyte archegonium and male gametophyte antheridium. Within the antheridium, the antherozoids are produced, and within the archegonium, egg cell is present. So antherozoids are released into the water and they reaches to the archegonium. So they fuses and they form the embryo. The embryo will develop and it form the sporophyte. So this type of life condition or life stages are called heteromorphic condition. They are differently, two phases are differently they are producing. Now after reproduction, the sporophyte is formed. When it comes to the sporophyte is maybe free living, or sometimes it is totally depend on the gametophyte. In case of Markensia, the Markensia sporophyte is totally depend on the gametophyte. When it comes to the Polytrichum and Funaria, their sporophyte is freely living. For observation of water only, they depend on the gametophyte. So two types of sporophytes we can find: completely dependent sporophyte and partially dependent sporophyte. So here the sporophyte is not freely living, but attached to the photosynthetic gametophyte. This is a sporophyte and it attached with the gametophyte. Okay, so why it is a not free living? Because to absorb the water and to prepare the food material, it do not have any specialized cells. And moreover, the archegonium is present in gametophore only. After formation of zygote, the zygote will develop on archegonium. So after development of zygote, it produces the sporophyte only. Okay, so that the sporophyte is a not free living stage. And within the sporophyte, now if we see, the sporophyte have differentiated into three stages: foot, seta, and capsule. So within the sporophyte, specialized cells are present. Within the capsule, specialized cells are present called sperm mother cells or spore mother cells. The cells are which present in the capsule, they are called spore mother cells. The spore mother cells, they undergone the reduction division and they produces the haploid spores. So when the haploid spores releases, they fall down on the earth, they produces the haploid gametophyte. That means the sporophyte is diploidic in condition and the gametophyte is haploidic in condition. Okay. So the spore germinate to produces the gametophyte. When they release it, when the spores are released, they fall down on the moist condition and they produces the gametophyte. So this kind of life cycle is called haplodiploidic life cycle. What is haplodiploidic life cycle? Haploid means a single set of chromosomes are present. Here, two sets of chromosomes are present. The gametophyte is haploidic in condition and the sporophyte is diploidic in condition. So what type of cycle is present? Haplodiploidic. Haplodiploidic life cycle is present in bryophytes. Now the total life cycle. So here the thallus, antheridium and archegonium is there. Here you can see the antherozoids. Generally the antherozoids bears two flagellae which are laterally attached. So it's a spiral-like structure. The antherozoids are spiral-like structures and they have two flagellas. They release it in the water and fuses with the archegonium. Now the embryo will form. Embryo developed on the archegonium. After development of embryo, it forms the sporophyte. Now the sporophyte is dependent on the gametophyte. So it's a sporophyte. Within the sporophyte, the spore mother cells are present. When the spore mother cells undergo reduction division or meiosis division, they release these spores. So here the spores are released. The spores are haploidic in condition. Now the spores will germinate and they produces the plant body or thallus. So this kind of life cycle is called haplodiploidic life cycle. Now the total stage is haploidic in condition, and here the total stages are diploidic in condition. Now the bryophytes have, based on their structure and other conditions, the bryophytes have divided into three classes. So they are divided into three classes: hepatic apsida, anthocerotopsida, and bryapsida. In hepatic apsida, generally the hepatic apsida classes plants are called liverworts. So the bryophytes have divided into three classes called liverworts, hornworts, and mosses. So now we'll see one of the class called liverworts. The liverworts belongs to the hepatic apsida. Generally, they are grown in moist or shady conditions. But Markensia is that one of the genus of Markensia. It can survive in desert conditions also. Mostly they are moist conditioned. When the moist condition is present, those region only the Markensia will grow up. This is the thallus of the Markensia, and some Markensia species are occurred in aquatic manner also. So mostly moist condition, but few plants are there, or few species are there which can survive in aquatic condition and desertic conditions also. The thallus is prostate and dorsiventral. Dorsal region and ventral region is there. At the dorsal region, we can, now this is the ventral region. On the ventral region, what you can find the gemma cups. These are the gemma cups, and you can find the midvein also. So the chlorophyllus or leaf-like structure present on the dorsal side, and at the ventral side, we can find the rhizoids also. So prostate and sometimes erect also present. The thallus exhibits the dorsiventral condition. Now, what kind of reproduction is happened? The reproduction generally done by the fragmentation, or the vegetative reproduction done by the fragmentation or gemma cups. Now, these are the gemma cups. So, gemma cups are, now how they are green in color, and they are multicellular asexual buds. So, they are green in color, they are multicellular, and they are asexual buds. That means when they detach from this thallus, when they detach and fall down on the soil, they produces new plant body. So the gemma cups. In liverwort, the asexual reproduction or the vegetative reproduction is done by the gemma cups. What are the gemma cups? The gemma cups are the green multicellular asexual buds. When they detach from the plant body, they produces new thallus. Now, sexual reproduction. The sexual reproduction is done by the antheridium and archegonium. We already learned what is antheridium. Antheridium is a male sex organ, and it produces biflagellated spores. Within the antheridium, the biflagellated spores are producing. And comes to the female reproductive organ, the female sex organ is called archegonium. So within the archegonium, a single ovum is present. It is the oogamy type of sexual reproduction is done in bryophytes. For their reproduction, they require the water. So how the sexual reproduction is done? When the antheridium, when the antheridium is developed, it produces the antherozoids. Antherozoids, generally they are biflagellated spores. Now here you can see the biflagellated spirally structures are present. So when the antheridium develops, it produces the biflagellated antherozoids. They release it in the water. So they swim in the water, and after that they reach the body of the archegonium. Now this is the archegonium one. Within the archegonium, ovum is developed. A single ovum is present, and 8 to 10 neck canal cells are present, because here the sex organs are multicellular. Okay, so in an archegonium, a single ovum is present. This is the ovum. If we just assume that it is an ovum, it's a single single cell ovum is present, and 8 to 12 neck cells also present. So when the antherozoids release it on the water, they swims and they reach it to the archegonium egg. So now here the antherozoids and egg fuses. So fusion of male and female spores or gametes is called fertilization. As a result of fertilization, the zygote is formed. Now where the zygote is located? The zygote is formed within the archegonium. Okay. So after formation of zygote, it do not undergone the meiotic division. So it develops, it forms the embryo, so that we can call the first embryophytic plants. So where the embryo is formed? The embryo is formed within the archegoniophore. The embryo will develop and it form the sporophyte. So that the sporophyte is partially or completely depend on the gametophyte only. Okay. So it is about the reproduction. Now, after reproduction, the zygote is formed, it develops into the embryo and it forms sporophyte. No, the sporophyte is placed on the archegoniophore. Now, what is the structure of the sporophyte? See the sporophyte. The sporophyte is differentiated into food, seta, and capsule. We already see that it totally or partially depend on the gametophore. So this is a gametophore. On the gametophore, we can find the sporophyte. So if you see the structure of the sporophyte is having three different structures. Foot, with the help of the foot, it attached with the gametophyte. Seta is a stalk-like structure, and capsule. Within the capsule, you can find the spore mother cells. So here, within the capsule, the spore mother cells are present. The spore mother cells, they are the two different types of spore mother cells are present. The fertile spore mother cells are produces the spore cells, and the infertile or the sterile spore cells produces the elaters. Now, what are elaters? Elaters are nothing but they are sterile spore cells. Okay. So, two types of spore cells are producing. The spore mother cell produces the fertile cells, which when they release it in the atmosphere, they are the male condition. The infertile or the sterile spore cells are called elaters. So within the capsule, the elaters are present and some spore cells also present. Okay. So it is the structure of the sporophyte. Now, once again, if you recapitulate, the sporophyte is having three stages: foot, seta, and capsule. Within the capsule, the fertile spores and non-fertile spores are also present. The fertile spores are the male spores, and the non-fertile spores are called elaters. So the elaters, maybe you can find elaters or pseudo-elaters are also present. Now, what is the function of elaters? The elaters are useful for the dispersals of the spores, because the spores are now they are present at the mouth side. Now this is the cap side. Near to the mouth or near to the cap, the elaters are present. When the spores are developed, the elaters become burst, so that the caps will burst and the spores release it into the atmosphere. So the functioning of elaters they are useful for the disbursement of the spores. Now it is example of the Markensia. Now this is about the one of the class called liverwort. The another class of the bryophyte is hornwort. Why we call hornwort? Here the sporophyte is looking into the horn shape. Now this is the horn shape structure. This is a gametophyte and this is a sporophyte. The sporophyte is horn shape. So this class is known as hornwort. In hornwort we can see if you see their general structure, the plant body is thalloid. Now this is thalloid. It also having the prostate and dorsiventral. Now this is the dorsal region. At the ventral region, the rhizoids are present. So the sporophyte is horn-like. This is a sporophyte which looks in horn-like. Again, the sporophyte bears foot, seta, and capsule. So with foot, instead of seta, you can see in intercalary meristematic zone. So what is intercalary meristematic zone? Here, if you see the structure of the sporophyte, now this is the foot, seta, and it's a intercalary meristematic zone. So the intercalary meristematic zone. Within the after intercalary meristematic zone, the capsule is present. Within the capsule, you can find the pseudo-elaters. Now you can see the red like structure, they are called pseudo-elaters. In liverwort, we can find the elaters. And in hornwort, we find the pseudo-elaters. Now what is their function? They are useful for the disbursement of the spores. They are steroid. They are the steroid spores. So the hornwort, in hornwort, the sporophyte is having the foot, intercalary meristematic zone, and capsule. Now this is the structure of the capsule. Within the capsule, we can find the pseudo-elaters. So example for hornworts are anthoceras. Now reproduction is also same. Sexual and asexual reproduction is done. In sexual reproduction, again, oogamous reproduction is there, antheridium and archegonium, as we already learned in liverwort. So this is about the second class, hornwort. Now we'll see the third class, mosses. Generally, the class of Bryopsida. The Bryopsida plants are called mosses. So in mosses, if you say their general cycle, the life cycle is the important or dominated stage is gametophyte. In all bryophyte, the dominant stage is gametophyte. The partially dependent stage is sporophyte. Now the gametophyte, it is developed into two stages. The first stage is called protonema stage. The second stage is called leafy stage or developed stage. When the spores released and they fall down on the soil or the surface or the moist soil condition, the spores will produce the protonema. Now, how the protonema? The protonema is a creeping green branch. Creeping means now they are erected with the soil. They travel with the soil. Creeping green and branched like structures having found in protonema. Protonema is the first stage. It is not developed stage. It do not bear any sex organs. So when the spores are released, when they form, when they develop, they produces the first stage called protonema. The protonema is a creeping branched condition which do not bear any sex organ. Now after development of the protonema, it converts or it produces the second stage. The second stage is called leafy stage. The leafy stage, in another word, we can call the gametophyte also. So this is the protonema stage. The protonema will develop and it form the second stage, which we can call the gametophore. So the gametophore are generally present at the lateral buds of this is the lateral bud. The lateral bud of the protonema will produces the gametophore. So here you can see the gametophore at the lateral bud. So the lateral bud of the this is the protonema. So lateral bud of the protonema will produces the gametophore. And after development, the gametophore bears the sporophyte also. So what is the structure of the gametophyte? The gametophore consists of upright, slender axis. Now how they are? They consist upright, slender, spirally arranged leaves. So here you can see the leaves. The leaves are spirally arranged. So the gametophyte is slender, upright, and it bears these spirally arranged leaves. And it also have the rhizoids. The functioning of rhizoids are to attach with the substratum and to absorb the water from the soil. And they also bear the sex organ. The protonema stage do not bear the sex organ. What are the sex organ? Antheridium and archegonium. So the antheridium and archegonium are present in the gametophore stage only. How the gametophore stage develop? The lateral bud of the protonema. The lateral bud of the protonema develops and it forms the gametophore. After development of the gametophore, they produces the sporophyte. So the sexual organs are present in the gametophore. The sexual organs are absent in the protonema stage. So here you can find two stages. The spores will develop and they produces the first stage of thalloid called protonema, and the second stage of thalloid is called gametophore, which is the developed one and bears the sex organs. Now, when it comes to the reproduction, the reproduction in mosses, the vegetative reproduction is done by the fragmentation. So we already learned what is fragmentation. And sexual reproduction, again, antheridium and archegonium is present. Within the antheridium and archegonium, the antherozoids and egg cells are present. So the antherozoids fuses with the egg cell and the fertilization is occur. The zygote will develop and the embryo also form. After development of embryo, the sporophyte is formed. Now the sporophyte, again, it have the foot, seta and capsule. So foot is inverted or inserted into the gametophyte. This is the seta and capsule. So sporophytic condition. In bryophyte, the mosses are well developed in liverwort. Then the liverwort or hornwort. So the mosses are well developed when compared with the other two classes. The life cycle of mosses. So you can see the protonema stage. So the spores are released and it produces the protonema stage. The protonema will develop and it converted into the gametophyte. So the gametophyte will develop the sporophytic condition. Now when the capsule releases the spores again divided into the protonema. So two types of developmental stages we can find, protonema and gametophore stage in mosses. If you see the capsule or the sporophyte bears the three conditions, foot, seta and capsule. Within the capsule, now this is the capsule and seta and foot is produced inside. It is inserted into the gametophore. So when it comes to the sporophyte, it is a capsule stage. Within the capsule, you can find 16 to 32 peristomial teeth. Now these are the peristomial teeth. The importance of peristomial teeth is to release the spore mother cells or to release the spores. The peristomial teeth are useful. So within the capsule, you can find spore mother cells. The spore mother cells undergone the meiotic division and they produce as the spores. So spores are present in the capsule. So when the spores are developed, the peristomial teeth will open and the spores will disperse into the air and they fall down on the soil and they produce as the they produce as the gametophyte. Okay. So the common example for the mosses are funaria. Now this is the funaria. Generally the funaria is called curd moss and polytrichum. The polytrichum is called hair camp moss. This is the polytrichum. And sphagnum. Sphagnum is also known as peat moss. This is a sphagnum. So three classes we have completed in bryophytes. Bryophyte we have the three classes, the liverwort, hornwort and mosses. Now with this we completed the bryophyte session. Now we'll go for the another one, pteridophytes. So what are the general characters of the pteridophytes? Generally the pteridophytes are the vasculated plants, vascular or tracheophytic plant, because the vascular system first time developed in the pteridophytes. So what are vascular system? Vascular system consists or having the xylem and phloem. The first time they are having the xylem and phloem, so that if you see the condition of the phloem is not so developed. The companion cells and the CV tube cells are absent in pteridophytes. So the phloem is not so developed, but at the first time the xylem and phloem is observed in pteridophyte, so that these are called vascular cryptogamous plants. So the pteridophytes are the first terrestrial plant which possess the vascular tissue, and they are the embryophytic. The embryo is developed embryophytic and archegoniate vascular cryptogams. Okay. So the pteridophytes, where if you see their habitation, the pteridophytes also observed in cool conditions, dampy or shady places. They also require water for their reproduction also. In pteridophyte, we can see the club mosses. Now this is the club moss plant and horsetail plants. So horsetail plants, this is the horsetail plants and ferns. Ferns generally they are having the macrophytes. The leaves are very large in condition. So the pteridophytes include club mosses, horsetails and ferns. Now if we see the general characters of pteridophytes, the main plant body of pteridophyte is called sporophyte. In bryophyte, the main plant body is gametophyte. But when it comes to this pteridophyte, the main plant body is sporophyte. So children, remember, the main plant body in bryophyte is gametophyte, which is haploidic in condition. But in pteridophyte, the main plant body or the dominating stage is sporophyte, which is diploidic in condition. And the sporophyte is differentiated into root, stem, and leaves. So here you can see the roots, the adventitious roots or the fibrous roots are developed. In the root, stem, and leaves, the true roots, two stems, two leaves are absent in bryophytes. They have the rhizoids. Instead of roots, they have the rhizoids, colloids, and phylloids. But when it comes to the pteridophyte, we can call root, stem, and leaf. So the root system, when it comes to the root system is adventitious root system. The stem is it is also erected and branched one. So on the stem, you can find two types of branches. So here, some of the branches having the leaves. So these are the leaves, and they do not have the sex organs. They do not have the cones. So this kind of branches are called infertile branches. The another branches bears only the sex organs or cones. So they are the fertile branches. So two types of branches are present. One is non-fertile or the sterile branch. We can see sterile stem. Sterile stem means here the cones are absent or the sex organs are absent. Now these are the fertile stem. On the fertile stem you can see the cone. So two types of branches are producing. Non-fertile or the sterile stem and the fertile stem. Now the leaves in pteridophyte, there are two types of leaves are present. Some are small in condition. The leaves are small in condition. Here you can see the smaller leaves. If the pteridophyte is having small leaves, they call microphyll. Micro means small, phyll means leaf. So small or scaly leaves are present. They are called microphyllous. And in case most of the pteridophytes are having the microphyllous. In case of Selaginella, it having the large, okay, sorry, the microphylls are present in Selaginella. It is an example of the Selaginella. In Selaginella, the microphyll leaves are present. That means the small leaves are present. When it comes to the fern, in fern, you can see the larger leaves. So this is the sphagnum. So in sphagnum, you can see the larger leaves also. So here is a fern. How the leaf is? The leaf is large in condition. So smaller leaves are present and larger leaves are also present. The smaller leaves are called microphylls. The microphylls are present in Selaginella. The larger leaves are called macrophylls. The macrophylls example are sphagnum and fern. So the root and stem, two types of stems are present, the fertile stem and non-fertile stem. So fertile and non-fertile stems are present, and the two types of leaves also present, microphyll and macrophylls. Now comes to the TS of internally shows the epidermis. So when you cut the stem with the help of the blade, if you cut the stem, you can find these structures because they are the first vascular cryptogams. So the vascular are the connective tissue. Xylem and phloem is developed. So this is the developed or the dicotyledonous stem TS. Okay.
హలో సార్ ఆయన వెళ్ళిపోయినట్టున్నారు, ఏమన్నా కనెక్టివిటీ ఇష్యూ ఏమో అయిపోయిన ఇంకొంచెం ఉంది కదా. వచ్చారులేండి, వచ్చారు మళ్ళీ వచ్చారు. ఓకే 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 సార్.
Madam, now can you see my screen?
Yeah, yes sir. Please continue.
Interruption was happening. Sorry for. Yeah. So the stem having the epidermis, cortex, and stele. This region is called stellar region. So within the stellar region, you can find the xylem and phloem and medulla also. So epidermis is the outermost layer. This outermost layer is called epidermis. It is a single layer. And cortex. The other region is called cortex region. Up to from epidermis to stellar region, the total region is called cortex region. And stele. Within the stele, now the vascular tissue is present. Xylem and phloem is present. So this kind of total tissue together called stellar region. This is the stellar region, and this one is the cortex region, and this one is the epidermis. So in higher developed plants, if we cut the stem exhibits or shows the epidermis region, cortex region, and stele region also. In pteridophytes also, the stem shows these three regions. But the difference is in pteridophytes, the stellar region is not so developed. The stele may be called protostele. So what is protostele? Protostele means the medullated, in the middle region, the xylem is present. X means xylem. So functioning of xylem is to transport the water and mineral. And the phloem, peripherally, the phloem region is present. The functioning of phloem region is to transport the food material. So in the middle, xylem region is present, and peripherally, the phloem region is present. This kind of stele is called protostele. So we can define protostele as the core part or the central part having the xylem tissue. Are present. So, sir, disturbance is happening. So in pteridophytes, we can find stellar region or different types of stellar regions are present. The main stele is protostele. Protostele means in the middle place, xylem is present and it is surrounded by the phloem. And you can find the siphonostele also. Within the siphonostele, the medulla is present. And solenostele, in the solenostele, we can find the leaves, gappages. And dictyostele, in the dictyostele, we can see many gappages are present. Now, when it comes to the reproductive structure, the sporophyte bears leaf-like appendages called sporophyll. Now, sporophyll, fill means leaf. Okay, fill means leaf. On the leaf, the spores are present. So, this kind of leaf is called sporophyll. On the sporophyll, you can find the yellowish color sorus or soren, Single one is sorus. The multiple organ are called sore. So on the leaf, we can find the sora. Within the sorus, we can find the spores. So the sporangia produces these spores by meiosis. How the sporangia is developed? The sporangia is developed by the sporangia may bear the two types of spores. It may produces one homospores, or it may produces heterospores also. What is homospores? Homospores means similar types of spores are producing. If the spores are same in size and structure, that is called homospores. If it is produces different types of spores, it is called heterospores. Example, here in Selaginella, you can find heterospores. See, the larger single spores are called megaspore. Now, this is the megasporangium. Within the megasporangium, megaspore is present. Megaspore is nothing but the egg. Okay. So how the egg shape is, it is very larger sized egg is present. And when it comes to the microsporangium, within the microsporangium, the male microspores are present, and there are many are present. Many small male microspores are they resembles the male structure. Megaspore resembles the female structure. So here the larger megaspore and the smaller microspores are present. Two types of spores are producing, so that it is called heterospores. So the spores development may be homosporic development or maybe heterosporic development, we can find it. Now, how this sporangium is developed? The sporangium is development in two patterns, that is leptosporangiate method and eusporangiate method. What is leptosporangiate method? A single superficial cell will initiate the development of the sporangium. It undergone development and it produces the total sporangium. So from the single cell or from one cell, the total sporangium form. So this kind of sporangiate development is called leptosporangiate development. So in leptosporangiate development, a single superficial cell will initiate the formation of the total sporangium. Now, when it comes to the eusporangiate development, in eusporangiate development, a series of cells or number of cells, here you can find number of cells. So, number of superficial cells will initiate the formation of the sporangium. So, many cells will initiate the formation of the sporangium. So, this kind of development is called eusporangiate development. So, in pteridophytes, we can find two types of sporangium development, leptosporangiate and eusporangiate. In leptosporangiate, only one cell will initiate the development of the sporangium. And when it comes to the eusporangiate development, many cells will initiate it. So two types of sporangial development will form. Now in some cases, the sporophylls may bear the cone-shaped structures. Now here the cones. So the cones having the reproductive parts. They may call cones or strobili. So you can find many cones are strobiles also. So here the spores germinated in the strobili or cones, they release it and they produces the gametophore. So within the spori or within the strobili or within the cone, the spores will develop. When they releases, they produces the gametophyte. So the gametophyte is also photosynthetic and free living. So in bryophyte, the sporophyte is not a free living one, but here the two things are living one. In pteridophyte, the sporophyte is also independently living and the gametophyte is also independently living. Now when it comes to the gametophyte structure, the gametophyte having the male and female sex organs, antheridium and archegonium. It also having the rhizoids. With the help of the rhizoids, it attached with the substratum and it can develop metis. And the sex organs, it bears the sex organs, which is called archegonium and antheridium. And these organs are multicellular and jacketed. They were and sessile. Sessile means they won't have these a stalk-like structure. So in gametophyte, it is also free living. It produces the rhizoids and the reproductive organs also produce antheridium and archegonium. They are multicellular reproductive organs are present. When the antheridium develops, it produces the antherozoites, and when archegonium develops, it produces the egg cell. So antheridium and egg, when they fuses, it forms the zygote. So this kind of oogamy is called zooidogamous oogamy. The female gametophyte is retained on the parent sporophyta here for variable time. So that means here it can live for some time for development of it. When it is developed, it becomes completely developed into the sporophyte. So after development of the sporophyte, the spores will release it, and before the releasement of spores, the embryo is formed. The embryo is forming, embryo is developing, and it develop into the sporophyte. Within the sporophyte, the spores are present. The spores undergone the meiotic division, and they produces the gametophyte. So this kind of life cycle. If you see the life cycle of the pteridophyte, here it is the main plant body is sporophyte. It releases the spores. It produces the gametophyte. Now the gametophyte is developing. It produces the antheridium and archegonium. And when they fuses, again they form the sporophytic development. So here also two types of phases we can find it: sporophytic phase and the gametophytic phase. Two different kinds of phases or stages are producing here. Now, one of the important fern is Diapteris. Now, this is the image of the Diapteris. Commonly, it is called fern. It have the root present in the underground, underground rhizome. So, from the rhizome, they produces the root. If you see the leaves are pinnately compound leaf. Pinnately compound leaf means, if you see the example of neem, in neem plant, from the midwing two branches are producing. So that kind of arrangement is called pinnately compound leaf. So from the midwing, at the branches, two branches are producing. So you can see two branches. So it is called pinnately compound leaves. And about this compound leaf structure, you can see, so the leaf shows the succinate venation. Now, what is succinate venation? In succinate venation, the leaves are initially they are coiled. The coiled structure type of venation is found in pteridophyte. So this kind of venation or this kind of arrangement is called succinate venation. The coiled leaf. The leaf is coiled. No, here it is a leaf. It is coiled in structure. So if it is coiled or it is spiraled, it is called succinate venation. And it comes to the petiole. The petiole is a multicellular hair. On the petiole, you can see the brownish hair-like structure. So here, if possible, you can find a hair-like structure. They are brown in color. These hairs are called ramenta. And on the leaf, you can find the yellowish color. No, the yellowish color is sporangium. And this is the sporangium. The sporangium is developed or surrounded by the another protective layer. This protective layer is called inducium. So the sporangium, now this is the sorus. Now the sorus is covered by the membrane structure. This membrane is called inducium. So when sporangium, within the sporangium, the sorus are present. The sorus is yellow in structure. It is covered by or surrounded by a protective layer or protective membrane, which is called inducium. Inducium is also present. So it is the fern structure. Now if you see the classification of pteridophytes based on few characters, the pteridophytes have classified into 4 classes. One is Coelopsida, example Coelotum, and another one is Lycopsida, example for Lycopsida are Coelogenella and Lycopodium. Then Spinopsida, Equisetum, and Pteropsida. Generally, the Pteropsida masses are present. Diapteris, Adiantum, and Pteris, these are belongs to the classes of Pteropsida. So Pteridophyta have classified into 4 classes, and we have seen the examples of these classes also. So with this, we completed the Bryophyte and Pteridophyte. So thank you so much for listening me. So I especially thanks to the coordinator, sir, and other principals also for giving this opportunity. Thank you so much, sir.
Thank you, sir. Excellent presentation, Satyanarayan garu. From the 5 subgroups of plant kingdom, you have extensively explained the characteristics, classes, and ecological importance of bryophytes and pteridophytes with excellent clear pictures, images as well. And students revisit the sessions, prepare notes, and practice diagrams also. Thank you once again, Satyanarayan Garu, for the wonderful presentation, and thanks to the subject coordinator, Dr. A. Venugopal Reddy, sir. Thank you.
Thank you so much. Thank you.